Hello, everyone.
What day is it?
The day before the midterm. That's the day that it is.
No, it's 2 days. 2 days before the midterm.
You don't want to have a midterm on Wednesday?
It's Monday still.
What's that?
It's Monday still.
It's Monday still?
It's 20 last day.
Yeah, but the midterm is on Wednesday. Right?
Correct.
Yeah.
We got you. You know when it is. We know
I say if it's not on Wednesday, I'm in trouble. No, that's not true. But midterm on Wednesday. So seating in here with respect to the midterm is difficult, as you can see, given the number of people. So what I ask people to do is come in first and sit every other as we've done before. So start here and then over, start here and then over, and then here and then over, and then there and then over. Then that will fill everything up. Okay, so we won't be able to sit everybody, every other person. So then you need to come to the front and sit. So if you're sitting next to somebody, it can be in the front, but not in the back. Make sense? So basically, when all the every others run out, then start filling from the front and moving backwards. Okay? This usually works reasonably well. But I will ask you, as always, to get seated quickly so we can get everything going. And there's a class after hours, so we need to make sure we get out of their way as quickly as we can. Okay? Let's see. What else? We have a review session I have scheduled. It's tomorrow evening. I usually like to do it tonight, but I'm teaching a class tonight at 5, and I can't do it tonight. But tomorrow evening at 5:15 in ALS 4001. Okay? 5:15 ALS 4001. And I will be there. And that's that. So the material that we will cover on the exam, I will let you know today where we will stop that material. Okay? So and then you'll know. So pretty straightforward. Okay. Let's see. 2 other brief announcements I've been meaning to make. One of which will I think definitely interest some of you, and that is I need to let everybody know that about the HHMI program. That is a summer undergraduate research program that I run at OSU, and I'm always looking for students interested in participating in that program. So if you are an undergraduate student and you are interested in doing research, the HHMI may be for you. It requires you to work with a professor and put together a proposal. The applications will come out a week from today. And if you would like to be on the notification list to get that application, please let me know. I'll make sure you get on the list. If you need help getting a professor or anything like that, let me know. I'll be happy to do that. And it requires you to be basically a freshman, sophomore, junior. If you are close to graduation, less likely that'll work for you. Okay? But freshman, sophomore, junior works fairly well. So if that's of interest to you, let me know. If we accept you in the program, we pay $4,000 to you for 11 weeks of work over the summer with a professor. So it's a pretty good deal. And the other thing is, if you want to come and laugh at your professor making a complete fool of himself, on February the 11th and 12th, I will be singing at the Majestic Theater with a group of other people. And we will actually sing one song of mine that you've heard before and a bunch of others there. So if you're interested in that, I'll post. I've got a bulletin of that posted outside my office, and you can come. My singing is fortunately a fairly small part of the evening. So, but anyway, if you want to come do that, it'll be a lot of fun. Okay. That said, we will now move forward and talk about, yes.
Do you know when the HHMI application is due yet?
Yes, the application for HHMI will be due typically on Tuesday of spring break. So we give you basically a couple of months to get it together, but I will caution you not to wait until then to apply. It'll be too late for you to be a very effective applier. I see several people in here have actually been in the HHMI before, and so you might talk to some of your friends in here. They may know about it. But if you have questions, come see me. Okay? Okay. So last time I got started rather hurriedly talking about cholesterol metabolism, and I forgot to basically tell you one thing. So I want to go back and tell you about that one thing here, and that actually is this guy here. This figure, which shows us how the very earliest molecules in cholesterol biosynthesis are made, includes this acetyl-acetyl-CoA, which I told you just a brief thing about, but I didn't tell you how we made this molecule. So it's important to know how we make aceto acetyl-CoA. There's an enzyme that we'll talk a lot more about later called thiolase. And thiolase turns out to be an enzyme that participates in two pathways. This is one of them. One of the pathways is it makes acetoacetyl-CoA. And the reason that it's able to participate in two pathways is it catalyzes the same reaction in both pathways, except for they're in reverse. So in the pathway here, it's making acetoacetyl-CoA. In the other pathway I'll be talking about next week, it's actually breaking it down. So thiolase is the critical enzyme for getting this whole process going, as we shall see. Okay. So last time I got very quickly up to this point. This point, I think, actually, this figure actually shows things fairly nicely. You may recall that after we make HMG-CoA, we decarboxylate it and create a class of molecules we call isoprenes. And isoprenes aren't specific structures, but they are 5-carbon-based structures. And these isoprenes give rise to a lot of things. One of the pathways that you're seeing here is the synthesis of cholesterol. And cholesterol is actually a precursor of steroid hormones. It's a precursor of bile acids. So this pathway ultimately gives rise to those. And there are side pathways here coming from geranyl pyrophosphate that can be used to make other lipids, including vitamin E and vitamin A. So there's actually 3 vitamins that can be made ultimately from this pathway: vitamin A, vitamin E, and as we shall see, cholesterol can itself be converted into vitamin D. So the 3 fat-soluble vitamins all can be made ultimately from this pathway. Anything that's made from this pathway is referred to as an isoprenoid. I-S-O-P-R-E-N-O-I-D. So an isoprenoid is something that's made ultimately from this pathway. Now, as I was going through rapidly the other day, I pointed out that the 2 isoprenes that we are following here is this one called dimethylallyl pyrophosphate that we decided to call Chelsea, I believe, right? And the other one we called IPP, isopentenyl pyrophosphate. We put Chelsea and isopentenyl pyrophosphate together. We create a geranyl pyrophosphate. This is simply a molecule that has 10 carbons. And we add another IPP to that, and we create a molecule that has 15 carbons. That's this farnesyl pyrophosphate. If we take 2 farnesyl pyrophosphates and put them together, we make a 30-carbon molecule called squalene. And squalene is a molecule I describe as the last linear intermediate. So this is the last molecule that we can identify clearly in the pathway that is basically linear. In the next step in the process of making cholesterol, we have to convert squalene, which is here, to this ring structure. And you can see there's an intermediate process. We won't worry about this intermediate, but the byproduct of this overall process is this guy right here that we call the first cyclic intermediate. And this first cyclic intermediate is known as lanosterol. Lanosterol, as I pointed out the other day, looks an awful lot like cholesterol. It has that characteristic 4-ring structure that we see in cholesterol. And the only main differences that we have between lanosterol and cholesterol are the decorations that are out here. And so we'll take a look at that in just a second. But lanosterol is obviously related to cholesterol. To go from lanosterol to cholesterol, as I said, it looks like it's fairly straightforward. This guy is here, this guy is here. As you can see on the figure, it's actually 19 steps to get there. Now, when I was a graduate student and I was learning metabolism for the first time, I had to memorize every one of those 18 steps and every one of those enzymes. And I always swore when I had to at that time, I would make my students do the same. But you see, I've mellowed as I've gotten older, thank goodness. But anyway, there are a lot of enzymes. There's a lot of energy necessary to do this. A lot of these steps require ATP energy and so forth to do these various conversions, moving of bonds, moving of methyl groups, and so forth. And as a consequence, this pathway of making cholesterol is very, very energy intensive. It takes a lot of energy to make cholesterol, and it's therefore very important that cholesterol participate in controlling its own synthesis. Thus, when cholesterol synthesis gets high, we want to have cholesterol participating in turning off its own synthesis. I'm going to show you a couple of ways in which that can happen. One way I've already talked about, that is the allosteric inhibition of HMG-CoA reductase. That was the enzyme that was inhibited by statins. It was also the enzyme that I said was the main regulatory enzyme for cholesterol synthesis. As we'll see in just a minute, cholesterol also helps control the transcription of enzymes responsible for making cholesterol. So there's two ways that cholesterol can, in fact, help to control its own synthesis. That regulation is shown in this figure, and it looks hairier than it really is. So let me tell you what's going on. What this figure is showing you is the way in which cholesterol is, by its absence, is turning on its synthesis. Okay? So if we have plenty of cholesterol, this process that you see on the screen will not occur. Okay? This process is ultimately making a protein called a transcription factor, and this transcription factor turns on enzymes necessary for making cholesterol. Okay? So now I'm going to repeat that. So this process, this pathway here is responsible for making a transcription factor that will cause enzymes for making cholesterol to be made. Now, it's kind of like a double negative here, because if we don't have cholesterol, we don't make this transcription factor, and therefore we don't turn on a synthesis of more cholesterol. So there's kind of a balance that's here in terms of having the cell control how many enzymes it has necessary to make cholesterol. Yes, Danielle.
You just, I want a little clarification. I think you just said if there's no cholesterol, this doesn't happen.
If there's no cholesterol, this does not happen. I'm sorry, if there's no cholesterol, this does happen. Sorry, if there is, oh, did I say that? Sorry. No wonder everybody's looking at me like, what are you saying? When there's plenty of cholesterol, this does not happen, because you don't want to have these enzymes if you have plenty of cholesterol. I apologize, yeah. All right, is that clear? If we have cholesterol, this process does not occur. Okay. Now, I want to tell you the steps in the process, and as I said, it's not overly complicated. If we look at this process, what we see is that there is this complex here called SREBP. It stands for, you don't even need to know what it stands for, but it stands for the steroid response element binding protein, blah, blah, blah. Okay? The SREBP is a protein that is embedded in the membrane of the endoplasmic reticulum. When cholesterol levels fall, this protein is transferred to the membrane of the Golgi apparatus. Okay? You may recall that the endoplasmic reticulum buds off to become the Golgi apparatus, so it's just simply a budding off that converts this into the Golgi apparatus. In the Golgi apparatus, this only happens when cholesterol levels fall. Okay? If cholesterol levels don't fall, it just stays right where it's at. If cholesterol levels fall, it moves to the Golgi apparatus. And in the Golgi apparatus, there's a serine protease that will clip this bond right here. Okay? The effect of clipping that bond is this guy is now free to go away. And it does. It goes away from this complex over here that protects it. It gets over here and gets exposed to another protease, a metalloprotease, that clips and releases the transcription factor. This is the transcription factor, a protein that activates transcription of genes. The genes it's activating are the genes involved in synthesizing, the enzymes involved in synthesizing cholesterol. Okay. So it's a several-step process. It only happens when cholesterol levels get low. Okay, everybody with me? You got a devious look on your face.
Well, what's the mechanism that makes it go to the Golgi? How does it respond to the
Well, so his question is, what's the mechanism that caused this to go to the Golgi? The movement of things from the Golgi, as we saw last term, is rather complicated in terms of cellular trafficking. So I won't go into. I don't have a way to go into that at this point. But suffice it to say that there are controls about which way things move in the cell. One of the ways we saw control of trafficking last term was we saw the trafficking that affected the insulin receptor going to the membrane. The gluts going to the membrane to let glucose in as a result of action of the insulin receptor. Okay, so trafficking controls where the proteins ultimately end in the cell. The controls involved in that are more than I'm going to go into here. Okay, make sense? Okay, I think that would be a good place to stop material for the exam. So let's stop it right there. And I'm going to be talking about the movement of cholesterol in the body, and that will be next, and that will be the material that we will start for the second exam. Yes, ma'am? Are we going to go into why it needs more enzyme? Yeah, it's a very good question. One of the things we'll talk later in the term about is basically the turning over of proteins. So cells, once they make an enzyme, that enzyme isn't there forever. There's a cycle through which they're broken down. Some proteins have a longer lifetime than others, but any protein, if you stop making it, eventually will disappear because of this turnover that's happening. So to answer your question, once we have that there, it's not there forever. So stopping its synthesis will, in fact, ultimately cause it to just disappear. Okay? I am referring to ubiquitination, yes. But that's another matter. Okay, this is an appropriate point for a song, and so I thought we might sing a song to help you remember cholesterol metabolism. You have to sing loud, you know the rules. Okay, this is to the song. This is to the tune of an old song called "When Johnny Comes Marching Home." Da 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 da. Right? It's called "To Make a Cholesterol." Please join me. Some things that you can build with acetyl-CoAs are joined together partly thanks to thiolase. They come together, 1, 2, 3, 6 carbons known as HMG, and you're on your way to make a cholesterol. To synthesize a mevalonate in the cell requires reducing HMG-CoA as well. The enzyme is a reductase controlled in allosteric ways when the cell's impelled to make a cholesterol. The mevalonate made in metabolic schemes gets decarboxylated down to isoprenes. They're linked together willy-nilly to build a pp-geranyl in the cell's routines to make a cholesterol. A single step links farnesyls, but that's not all. The squalene rearranges to lanosterol. From that, there's 19 steps to go before the sterols apropos, which you must recall to make a cholesterol. The regulation of the scheme's complex in ways inhibited by feedback of the reductase. And statins mimic, so they say, the look of HMG-CoA. So we sing their praise and not make cholesterol. Excellent. That's great singing, guys. Awesome. What's that? The first song you've known. I'm an old geezer. I have all these old songs. So, all right, you guys know I'm an old geezer. So, all right. Now, what I want to do now is talk about a process that generates probably as many questions as any topic I talk about in the class. Students find it very much of interest because it's something that's very much a topic that has touched many people. This topic is the movement of cholesterol and lipids in the body. And this is a fascinating topic. There are something like 7 Nobel Prizes that have been won in the history of the Nobel Prize for cholesterol. So if you want to talk about a compound that's interesting and a compound that we don't know all the stuff about, cholesterol is one of them. We know a heck of a lot more now than we did even 20 years ago. But even still, there's aspects of cholesterol and its movement in the body we don't understand. Okay? So I'll apologize in advance for some of the questions that you're going to ask me, and I will say we don't know that. okay? Because there are things that we don't know about this. But it's a fascinating topic. Well, the movement of lipids in the body is important, and it is complicated, okay? We start by talking about the complexes through which lipids move in the body. Now, lipids we're talking about in general here are all water insoluble. Water insoluble. This is the problem. The problem is that we've got things in our body that aren't dissolved in water, but our bloodstream is water. So if we're going to move them through our body, they've got to go through the bloodstream for the most part. And to do that, we've got to solubilize them. Well, solubilizing them with a detergent would be really nice and really easy, except for the fact that too much detergent is going to cause your proteins to denature, and we don't really want to do that either. Okay? So we've got to have a mechanism of moving these guys through the body. The body's mechanism that it uses is to create what are called lipoprotein complexes. Lipoprotein complexes. Now, these are very involved. You can see there's different proteins that are involved in these. And their names, some of their names are over here. Okay? And what they are proteins that on one side are fairly hydrophobic, so they can interact with, let's say, fat. And on the other side, they're hydrophilic, so they can interact with water. So to move these non-water soluble things in the body, we make a complex. We take a little fat globule and we basically surround it with these proteins, and that allows them to be dissolved in water and to move accordingly. Okay? Now, these start from guys right here that have the very lowest density as measured in grams per milliliter to those up that have the highest density. All right? The lowest density we can think of as big, fluffy complexes full of lipids. Now, when I say lipids, what am I talking about? Mostly I'm talking about fats, fat-soluble vitamins, and cholesterol. Three things that are very water or fairly water insoluble. Okay? So the body's got to move them from one location to another, and they package them up in these complexes. All right? Now, the first of these complexes, chylomicrons, appears not in the normal transporting process to control things, but rather as a function of what we eat. Okay? So what I'm going to do is take you through what happens when you eat a Big Mac and a Maxi Fries. Okay? And we're going to package that stuff up, take it through your digestive system, and then bring it back to your liver. And what you're going to see in this metabolic process is that this is the reason we have to think about two things with respect to cholesterol. Three things, actually. Okay? One, what we eat. Two, what we make, which we've already talked about. And three, what we store. All three of these are important in our overall cholesterol levels in our body. Okay? All right. So we start with what we eat. So we're going to go have that Big Mac, that Maxi Fries, and we said, please put on a little extra grease. Okay? And when we do that, we create, still not the thing I'm after here, cytosis, particle fates. Here's what I want. Okay. Now, I've just eaten a Big Mac. All right? The Big Mac gets to my intestines, and the intestines going, oh, wow, this guy has really done us a great favor today because he's eaten all kinds of fat, because we're going to be able to use this to make it through this hard winter, and we're going to take this fat and we're going to go store it away. Right? Okay? I like that sound effect. That's pretty good. I wish it sounded more hollow. That doesn't sound very... It sounds pretty solid. I don't know. It's not muscle, though, I can tell you that. So, all right. So, the intestine gets a hold of this stuff. Now, when the fat hits the intestine, okay, which is different than when something hits the fan, but when the fat hits the intestine, it may be like the same thing. I don't know. The fat hits the intestine's got to do something with it. Now, what the intestine does with it is really interesting, and it's kind of odd. And I'm going to help you to solve the mystery of why it does what it does. It'll grab a fat. A fat has a glycerol and 3 fatty acids on it. And it's got to move that fat across the intestinal wall so that it can ultimately get to the bloodstream. Okay? Notice I say ultimately. It goes first into the lymph, as we'll see. But But it's got to make it into the bloodstream, ultimately. All right? That means that that intestinal wall has got to pass the fat through itself. All right? That's not an easy thing to do. And so what this intestine does is there are enzymes there that will clip off 1 or even 2 of the fatty acids off of the fat, transport them and what remains of the fat across the intestinal wall. And when they get across the intestinal wall, they get repackaged. Okay? Now, does this sound a little odd? Take them off, move it across, and then go back and put the fatty acids back on there to remake the fat. Okay? Everybody understand what just happened? My question is, why do you think the body does that? What's the advantage of taking the fatty acid off and taking that across like that? Well, it's easier to get it across. Question? You have a comment? No?
In case there's any essential fatty acids.
In case what?
There's like essential fatty acids on that.
In case there's essential fatty acids, but it's taken off and it's put back on. So it's not like there's anything else that's happened to it. I want you to think, yes, sir?
Maybe it's balance of energy, like break it down, you get energy from that side and the other side.
That'd be a good thought, but that's not it, unfortunately. There's actually a more chemical reason why this happens. Okay? Think about what happens. Where do we see fatty acids out in the real world? What are fatty acids used for? For what? For soap, right? What do we use soap for? Solubilizing things that aren't soluble, right? This helps to solubilize that very insoluble molecule, all right? So it acts a little bit like a detergent. It's actually helping to solubilize those fat molecules that are there. That helps to get it across the lipid membrane. All right? Now, we don't want those fatty acids staying off there because we want to package this fat up and get it elsewhere. We also don't want this detergent going elsewhere and causing big problems. So we put the fatty acid back onto the glycerol molecule and remake that fat. When we get that fat across the intestine, all right, it's packaged up first into some complexes called the chylomicrons. So we follow going over to the right here from the intestine. We've gotten it across the intestinal wall, and it's packaged up in these lipoprotein complexes called chylomicrons. This happens in the lymph system of our body. Okay? The chylomicrons then now are big, fluffy complexes of lipids. This includes fats, fat-soluble vitamins, cholesterol, okay? And these proteins that help them to be soluble in the aqueous environment of the bloodstream. The chylomicrons leave the lymph system and enter the bloodstream. Okay? Now, I want you to think about these great, big, fluffy complexes, and I want you to be thinking about heart attacks. How many people have you ever heard who say they go to McDonald's and say, I'm going to have one of those and a bypass to go with it? Okay? People joke about that all the time because you think, oh, wow, here's what's going to happen. I'm going to eat one of these and I'm going to have a heart attack. Imagine these big, fluffy complexes, right? It turns out these have nothing to do with heart attacks. Fooled you, okay? Nothing to do with heart attacks here, okay? The reason that they have nothing to do with heart attacks is they pass into the bloodstream, and in the bloodstream, because they're so nice and big and fluffy, they go floating along to the capillaries and they get stuck. Okay? They get stuck. Nope, still not at the heart attack point here. Once they get stuck there, this is actually a target for the cells at that location to start pulling the goodies out of the chylomicron. So they secrete enzymes called lipases.
What's the date?
The cells in the capillaries are secreting an enzyme called lipases. And what lipases will do is they will start breaking down those fats that are in the chylomicron. Okay? So the fatty acids are being gobbled. The glycerol is being taken. This causes the chylomicron to start shrinking. Some of the fat-soluble vitamins may come along just for the ride. And the chylomicron gets smaller and smaller. That chylomicron getting smaller and smaller causes it to become small enough that it passes now through the capillaries. So the chylomicrons got stuck. They got the Oreo center taken away, for the most part. Notice I didn't say anything about cholesterol there. The cholesterol that was in there is untouched. It stays there. Yes, ma'am?
How fast does this happen?
How fast does this happen? I don't have an answer for you. It's happening, I would say, within minutes, but I can't tell you beyond that.
So, like, in that minute after we've eaten the Big Mac, we have our capillaries blocked?
Well, so this sounds like, oh my God, my capillaries are starving here, right?
Yeah.
Keep in mind that you have millions of capillaries. And so you're not completely blocking all of your capillaries, no. And you're not even necessarily completely blocking the blood flow in those capillaries, but it just happens to be stuck at a particular place. Okay. So I'm taking some artistic license here.
Had a little panic attack.
Panic attack. This will keep you away from a Big Mac. This is good. Okay. All right. So these guys shrink, and they pass now through the capillaries, and they get into the venous system, and they come back to the liver. And the liver says, yum, yum, and the liver gobbles them up. Okay? So that's what's happened in this dietary cholesterol, or these dietary lipids that we've just eaten. Okay? Some of them got removed. Some of the lipids got removed. The fatty acids, the fats, the fat-soluble vitamins, but not the cholesterol. So we can imagine that what we've done is we've basically enriched this lipid mixture with cholesterol. Okay? All right. So it gets back here to the liver. All right? Pulls it in. Now, the liver turns out to have a very, very important role in regulating how much cholesterol is in the body. And it uses a really interesting and odd system to do it. Okay? Now, in the normal scheme of things, what happens is the liver is sitting here, and it decides, okay, the body is needing some cholesterol. I'll tell you in a minute how it decides that. But it decides that the body is needing some cholesterol. So what it does is it packages up cholesterol into a lipoprotein complex called the VLDL. Very low density lipoproteins, a lipoprotein complex. VLDLs are big and fluffy, and they go out, and guess what happens? They get stuck again in various places, and the insides get digested. And yes, the VLDLs have the same things as the original: fat, fat-soluble vitamins, and cholesterol. The cells start pulling these things away, free fatty acids and so forth, and the VLDLs become smaller, and now at this point we call them IDLs, intermediate density lipoprotein complexes. Okay? These guys get chewed on, and when they get chewed on, every chewing that we're doing here is removing fats and fat-soluble vitamins. We're getting over here to LDLs, the low-density lipoproteins, and there's not a lot of other goodies there besides cholesterol. So we've been enriching again for cholesterol, cholesterol, cholesterol. The cholesterol concentration is highest right here. LDLs are what your doctor calls bad cholesterol, because high levels of LDLs are correlated with atherosclerosis, an increased chance of stroke. Okay? Why do we make LDLs? Well, it turns out we make LDLs as a way of delivering cholesterol to cells that need it. They're there for a purpose. Okay? One of the purposes is to deliver cholesterol to cells that need it. A few days ago, I talked about receptor-mediated endocytosis, and I said it was another way of getting things across the cell membranes. There were specific receptors that would bind to LDLs, and they will literally absorb them into the cell. The cells that need cholesterol will have receptors that are binding to LDLs and pulling the LDL in. Okay? Now, the LDL still has a little bit of fat and fatty acids and fat-soluble vitamins and so forth in there, but mostly the cell is trying to get the cholesterol in. Okay? Now, not all of the LDLs get taken up by the cells. It sort of makes sense that the cell is only going to take up an LDL if it needs cholesterol. If it doesn't need cholesterol, it's going to say, thanks, I'll pass, and lets the LDL go floating on by. Okay? The LDLs, if they don't get taken up by the cells, go right back to the liver. So the LDLs, if they don't get taken up, go back to the liver. The number of these that make it back to the liver tell the liver how much the cells need cholesterol. The more of them that make it back, the more the liver knows, whoa, those guys are full of cholesterol. I don't need to be putting more complexes out. Okay? If none of them make it back, then the liver says, whoa, those guys are hungry. I better package up some more VLDLs to feed those cells that are going to be needing this cholesterol. Make sense? Okay. Yes, sir?
So is cholesterol stored mostly in the liver?
Cholesterol is stored in the liver. It's stored in the cells. It's stored in a variety of places. So the cholesterol is also stored in virtually every cell in your body. Okay. So the cells have a certain need for cholesterol because you remember they want to put it in the membranes. Right? So they've got a need for that cholesterol, and when they need it, they're going to be grabbing it out of there. The most critical component of this system that I've just described to you for controlling cholesterol levels is right here. The liver has specific LDL receptors. When the LDLs come back to the liver just gobbles them up like it gobbled up the chylomicrons. Okay? It just gobbles it up like it gobbled the chylomicrons. The more receptors are getting LDLs, the more the liver knows, quit putting more stuff out. Okay? Now, that means we have an idea now about why some people may have high cholesterol, even if they're controlling how much cholesterol they're getting in their diet. Okay? I'll tell you a story about a student that was in this class about 8 years ago. She was a graduate student, and she came and she told me that her son had a rare genetic disease. And I was aware of the disease, and I said, no way. And she said, oh yeah. The disease is known as familial hypercholesterolemia. Okay? Familial hypercholesterolemia. Mouthful of the name, I'll put it in the highlights. Okay? Familial hypercholesterolemia is extraordinarily rare, and it is caused by, what it does is people who have this are unable to make LDL receptors in their liver. Oh, this is bad. Okay? It's really bad because the liver constantly thinks that the cells are taking everything up. So what's the liver doing? It's constantly putting out VLDLs, even though the LDL level is enormous. Now, to give you an idea, and it turned out that what she was telling me was absolutely true. Okay? To give you an idea, what's considered fairly high cholesterol is a reading of about 240 to 250. At that point, they start giving you medication to lower your cholesterol levels if you can't control it with diet. This kid was like 7 years old before they knew he had the disease. And he had on him sores that started appearing. Okay? And they looked like blisters. And they took it to all these doctors, and the doctors didn't know what it is. And finally, they looked at it and said, oh my God. It turned out the kid lacks these LDL receptors. They checked his cholesterol level, and his reading was over 700. He was 7 years old. Now, fortunately for this kid and for this family, there are cholesterol drugs now that control this. Okay? If he had been born 10 years earlier, he would have died. The average life expectancy of somebody with hypercholesterolemia is about 10 years old because they die of a heart attack because everything is just all plugged up. Okay? They gave this kid a set of drugs and so forth and got his cholesterol levels down to about 250. And from all the sound of all things, he's going to live a happy and healthy life. But an amazing story, and I think somewhere in here I may even have a picture of his sores on his leg, which I won't show you here. You can look at them if you want to. But an amazing story. So, this is a bizarre case. All right? If you completely lack receptors, what happens? Well, you lack receptors, you're not going to live very long if it's undiagnosed. All right? But what if your receptors are slightly off? What if your receptors just don't get the LDLs when they come back? Or maybe they get them, but they don't signal into the liver. What do you suppose is going to happen to that person? They're going to have higher LDL levels. Okay? When you hear stories about people that can go out and they can eat a dozen eggs for breakfast every day and all they want, and their cholesterol levels are still disgustingly low, it tells you they have a system that's working very well. The system is working as it should. Now, if you overload your liver, how could you overload your liver? Eating too much fat. Remember, liver is only got so much capacity to hold things. If you overload your liver and you have a weight problem, you may be overloading the ability of the liver to do anything. That is in terms of controlling things. Therefore, what may happen is, even though the signaling is going on, the liver says, hey, I can't handle it anymore. When you eat, I've got to put this stuff out because I've got no more place to put it. So one of the things they try to get you to do is control your diet. That's why they will try to get you to reduce your fat intake and reduce your cholesterol intake. If those things don't work, that's when they start giving you statins, which will stop the synthesis of cholesterol. And those do a pretty good number on knocking these levels down. Okay? There are other strategies. Cholesterol turns out to be recycled in your intestine. Okay? Normally, the only way that you get rid of cholesterol in your body is in your feces. Okay? And even at that level, it's not significant because your intestines are pretty good at recycling it and bringing it back, saying, hey, look what I brought back for you. Okay? Thanks a lot, right? Well, another strategy in lowering cholesterol is actually to lower the recycling so that you actually do excrete more and retain less and therefore lower your cholesterol levels. Okay, that usually stimulates a bunch of questions, so I'll stop there for the moment and take any questions you might have. Yes?
Does age reduce the ability of the LDL receptors to signal with the rest of the liver?
Does age reduce the ability of LDL receptors to signal with the rest of the liver? I don't know the answer to that question, but the higher cholesterol levels do tend to correlate with age, and you might suspect that there are some problems associated with that. You also think about things that people do to their livers over time, alcohol, and you also think of their weight. So both of those may play roles in that process. Yes?
So the drugs that kids is taking, does it help the body make LDL receptors or doesn't it?
Okay, so the drugs that the kid is taking, her question is, does it help the body to make LDL receptors in the liver? No. The kid is genetically unable to do that. All right? So basically what they're trying to do is starve the body for cholesterol. That is, stop making it as much as you possibly can. They watch his diet very carefully. He's also on the drugs that they stop the recycling. And even with that, his LDL levels are fairly high. Shannon?
What's the HDL?
Okay, so I haven't gotten to that. HDLs are what your doctor will... Well, let me ask for questions on this, and I'll come back to HDLs. So any other questions on what I've just told you? I haven't told you the good side of it yet. Yes, ma'am?
On the kid that have the disease,
Yep.
the source, cholesterol deposits?
Probably cholesterol deposits. Pretty disgusting. Yeah. They had no idea. They had no idea whatsoever. You know, it's a recessive disease because it takes defect of both genes in the receptor for you to be able to completely be missing this. So it's not very common, as I said. At that time, they were saying there was about a dozen people in the United States that had it. And when she told me that he had it, I thought, you're a little off, lady. And she showed me the stuff, and I go, oh my God, amazing. Yes, ma'am.
The drugs that keep the intestines from reabsorbing, is that the ally weight loss medication?
Is that the allied weight loss medication? I have no idea. No idea. There are drugs that will also stop fat reabsorptions, so I don't know the answer to that question. Sorry. Who? Oh. Oh. Back to biochemistry. Okay. Any other questions about this before I talk about HDLs? Now, yes?
Someone has a liver disease or something that knocks out use of their liver for short term or long term.
Yep.
Is that when they have to go off of eating fats and change their diet? Or how do
Yeah, if a person has liver disease, is that why they control their diet to some extent? And the answer is yes. Liver, as you can imagine, plays very, very important roles. You saw it last term in terms of its levels in regulating glucose in the body. Now you see it's controlling cholesterol in the body. You also see sort of indirectly here it's controlling fat in the body. So when they're controlling diet, they're really trying to give the liver as much of a break as they can in handling all these things. So yes, the answer is...
they still get proper nutrition?
What's that?
Do they still get proper nutrition without?
Well, they have to get proper nutrition to stay alive, obviously. So the answer is yes, they do. But they're going to be very careful in terms of how much they're giving the liver. And my suspicion is, and I'm not a physician, so I can't tell you this from a medical perspective, but my take on this would be they're probably going to be prescribing things that are a minimal level necessary to survive, basically. So you're probably going to have a fairly restricted diet, a fairly low-calorie diet, because you want to get all these other, less for the liver to have to process. Okay, yes?
So LDL isn't really bad cholesterol, it carries cholesterol.
That's right. So, and it's not even bad cholesterol per se, because LDLs are there for a purpose. But they've achieved that name because high levels of LDL correlate with atherosclerosis. I'm going to tell you how that happens before I tell you about HDLs, but that's what I'm getting to here. Okay? Why do high levels of HDLs correspond to more atherosclerosis and more stroke? Okay? The answer has to do with what happens to LDLs. It doesn't happen to the other complexes for the most part. Okay? What happens to LDLs is they're very susceptible to oxidation. Now we see reactive oxygen species coming into the front again. They're very susceptible to oxidation. When they get oxidized, okay, they create free radicals. Those free radicals may cause them to become attached, for example, to the arterial wall. Okay? Remember, whenever we have free radicals, we have all kinds of unexpected reactions that can happen. When that happens, the body looks at this thing that's stuck to its arterial wall and it says, whoa, problem, we got to deal with a problem. And how do we fight a problem? We fight a problem with our immune system and we attack it with macrophages. That's how we get rid of infection all the time. This isn't an infection. This is a complex that's been there, and the more complexes we have, the more likely we're going to have these things getting stuck. Okay? The body attacks it, and it attacks it so efficiently that it creates a big foamy mass. This may attract, in the process, other LDLs there, because these macrophages are recognizing this LDL here that's a problem. Okay? So now we've got this complex of macrophages, LDLs. It's making what is the beginning of what we call an atherosclerotic plaque. An atherosclerotic plaque is a block. If it gets big enough, it will, in fact, stop the flow of blood. If this happens in your heart, you have a heart attack. If this happens in your brain, you have a stroke. The more LDLs you have, the more likely you will have this problem. Okay? How do you get more LDLs and why, and what's more likely to have this problem? Well, one of the ways that you increase your concentration of reactive species in your bloodstream is by smoking. Smoking will increase the number of reactive species present in your bloodstream. Antioxidants do not appear to protect against that. How do you reduce the level of reactive oxygen species? I couldn't say the word. Reactive oxygen species in your bloodstream? Exercise. Polyunsaturated fats. Those two things appear to reduce levels of reactive oxygen species. Stopping smoking obviously does as well. And these two things also appear to help favor an increase in the concentration of the good cholesterol known as HDLs. HDL stands for high-density lipoproteins. And their role is rather complicated. I'm not going to specifically go into it here, but suffice it to say that HDLs recognize fragments of LDLs. They recognize cholesterol floating around freely, and they grab it up and haul it back to the liver. Your doctor will call HDLs good cholesterol because they do appear to scavenge pieces of cholesterol and LDL that would otherwise cause problems. So your doctor wants to see your HDL levels to be high and your LDL levels to be low. And as I said, HDL levels go up with exercise and proper diet. They go down with smoking. Okay? That is a good stopping point. Let's stop there.
Friday's video? The link wasn't there as of this morning.
I put it up over the weekend. Should be. I'll look and
It was
see,
all
but
black.
it should be there. Really?
Yeah.
I'll look.
Okay, thanks.
Good luck.
Hey, I have a quick question.
How's it going, John?
Good, how are you?
Good. What's up?
Hey, so why, I don't understand why the concentration of cholesterol in LDLs is higher than VLDLs. It seems like in VLDLs you have a lot of cholesterol and then there's just a smaller amount. Why is the concentration different?
Nothing is taking the cholesterol.